filtering in so I can just make some comments. I'm almost done grading the quizzes. I plan to have them back tomorrow for discussion. A bunch of things that we scheduled this morning, so if you, that's on your radar, make sure to stop by October, stop by Almine Hall at 10 o'clock, the fourth floor. That's what we'll be doing the quizzes. Pretty good results on the quiz. I think having that extra practice problem helps. Still graded kind of liberally, but think about, the goal with doing the quiz and then giving back the key of the quiz to students is that you can think about what you do and do not still know about linkage, because that will be on the exam. So what will they cover? They'll cover pedigree. So chapter 6, linkage, chapter 7, it'll cover chromosomal mutations that we did in class, like inversion mutation. It'll cover transcription, translation, so what we're doing this week, and then last week, DNA replication. So there is a study guide available. It should be posted with the discussion problems for tomorrow. So if you have a chance, you can start looking at that and asking questions about, well, how would you do this? Whatever material we don't finish this week, we'll do Thursday after spring break. And if we cover all the material, great, we can just do kind of like an in-class study session since we'll miss that for discussion. Any questions on that before we get? I think it's like Tuesday, March 22nd. Does that sound right to people?
Yeah.
So spring break, Thursday lecture, the week after, Tuesday, and in-class exam. Any other questions? Okay. So today's lecture is mostly just to try to summarize the information that's been in the reading and highlight what I think is important, and provide context for what we're thinking about when we're talking about the genetics or the core genetic aspects of DNA transcription or replication and then transcription and translation. So this is the slide we stopped at last class, and at the beginning part of transcription. And so there were some really key aspects of this, is that RNA molecules are synthesized, anti-parallel. So what does that imply? It implies that we have to know something about the polarity of the template strand of the DNA to be able to understand the direction that an RNA takes. So that's always in the 5 to 3' direction. So that's similar to DNA transcription. The chemical nature of the nucleotide and the nucleic acids, the polymers themselves dictate how the process is going to work through RNA polymerase. So this was a clicker question. We're not going to redo it again, but we're going to think about it. So it was kind of 50-50. So the question was, here's a stretch of DNA. It's to highlight two things. One, transcription is not just occurring linearly along the whole stretch of DNA. It's occurring in specific regions, specific times to make a product, right? To make an mRNA that may or may not become a protein. Not all genes are in the same orientation. Genes can be in different orientations. And when that occurs, it means something. It means that sometimes the top or bottom strand will use this template. There's no universal template strand. It's really dependent on the gene and the orientation of the DNA you're looking at. So the question was, which strand is the template for gene B? So gene B is that yellow one in the middle. So what would be a good place to start to answer this question? So we left it off on top at 50-50. Some people were saying top, some people were saying bottom. So what would be our important place to start? There's many ways, but what's one idea?
Five prime?
Yeah, you have to identify the 5' of something. Yeah. So you can start at the DNA or RNA. If it gives you the polarity of the DNA here, we can write it closer. So this would be 5', right? I'm just transcribing what it has there. The RNA has to be anti-parallel. So if this green thing this green arrow can mean two things. It can mean the way that the RNA polymerase is traveling. It can give us the directionality of the RNA itself. Either way, it's telling us the same polarity of that molecule. So which two strands, so we have our RNA, I'm giving you the polarity of the RNA now, we have two DNA strands. Which DNA strand is anti-parallel to the RNA strand?
The top one.
The top. So here, let's answer which one is the template. The top is the template. So you should be comfortable doing this from lots of different contexts. This anti-parallel is a fundamental aspect. It's going to get us to translation too. There's going to be another molecule, a tRNA molecule, that's going to be anti-parallel to the messenger RNA. That tRNA is carrying the amino acid that's important for translation. So this fundamental principle of anti-parallel, you're going to see over and over and over again. Any questions on that? And again, why is that happening? Why are we calling that the template? What does that signify? It signifies that this nucleotide are always going to be added in this 5' to 3' polarity. So as this RNA strand grows, which way is the polymerase moving? It's moving towards the right? It's adding nucleotides so that 3' height drops, so that it's moving 5 to 3, making those phosphodiester bonds. Okay, so similar to transcription, there are three main steps replication, there are three main steps. There's initiation, elongation, termination. These don't look exactly the same for prokaryotes and eukaryotes. We're going to go through a couple of prokaryotic examples just because they highlight some fundamental aspects, but the general principles are similar. So we did this already for replication. What were some of the features that we saw in any of the classes? I'll add them as you shout them out to me. What were some things we needed for initiation? Or elongation or termination?
Enzymes?
Yeah, we need the proteins to do the work. Yeah. So what's our protein for elongation? For DNA replication, yes. For RNA transcription, it's RNA polymerase. So again, the ASE suffix ACE means an enzyme, and then you look ahead and it tells you the job. So this enzyme is making an RNA polymerase. Yep, great. Thank you guys for that. Any other things you can start filling in? So in replication, we had an origin. What part was the origin important for?
Is it for the fork?
Yeah, so the origin was important for initiation. So we don't call it an origin for transcription, we call it a promoter. But it's the same idea.
Yeah.
It's going to be a specific DNA sequence. And it's going to have some protein interaction. So we have, how are we going to recognize where to start? We're going to use a promoter that's going to contain some other sequences as well that are going to increase transcription. How are we going to continue building this polymer? We're going to do it through an enzyme, RNA polymerase. How are we going to finish? How did we finish DNA replication?
Codon.
Bubbles kind of met, and we end up with this really long linear strand. Here we have to stop. We can't go all the way through. We don't want to transcribe all genes simultaneously. We want to transcribe the specific gene we care about in the specific context. So we actually need another specific DNA sequence to tell us when to stop. And that's going to undergo DNA-protein interaction. So just to draw on the kind of parallels and differences between replication and transcription. So these are some of the regulatory sequences. So they're regulating, they're promoting. So we have, again, that template, non-template strand, but now adding some details, not all of it, some. So we have a promoter, right? That's where there's going to be a transcription factor binding directly to this type of DNA. We have the coding region, the part that will actually be the protein that sequentially, and then the termination sequence. Not all of that gets included into the mature mRNA, so that's what's shown at the bottom, but it's really important for initiation and termination. Right, so we just said the promoter is for initiation. Start of transcription. Not translation. We'll see later why that's the case. And then we have the terminator, which already is labeled as the transcription stop site. So what does that do? It causes transcription to occur at a really defined location. So the next slide is going to give an example of a very specific way that a protein is going to interact with the promoter in E. coli. So you do not have to memorize this. It's not universal. This is a very prokaryotic thing, but the idea is that there's a specific protein DNA interaction. So this is RNA polymerase in E. coli. There's a lot of subunits. You don't need to know all the subunits, but hopefully you can kind of see in that three-dimensional picture. It looks like there's lots of pieces. The one that's really important for initiation is the sigma factor. So that's kind of in the lighter yellow color. So sigma factor is going to directly interact. It's necessary for initiation in prokaryotes. So in this figure, right, you have the promoter is the kind of yellow chunk of DNA. It's directly binding in what they call that close complex with the sigma factor kind of in the second line down. So this is initiation. That sigma factor is not required for all of elongation. At the end, right, it's disassociated down here. So you do not have to memorize this, because this is not what is occurring in eukaryotic genomes, but the important part is initiation has to be initiated by a DNA RNA sequence interaction. So that's what this is showing in your book. So if you've read all the details in your book, great. But it's just to try to get you to think about how DNA and protein are required for initiation. So that sigma is very specific for prokaryotes. The broader term that we have often for proteins that are going to initiate transcription or are essential for transcription are transcription factors. So these control the rate of transcription. In DNA replication, there's only so many parts of the cell cycle that you're replicating your DNA, right, in S phase. Transcription is happening at different times in different parts of an organism. So the main thing here is that you want to control that, and you can do that through transcription factors. So some bind directly to the promoter. Others recognize regulatory sequence. By recognize, it's really binding. Remember, it's a DNA binding protein. So in prokaryotes, we have kind of this limited promoter, and we'll see how that's organized kind of after the exam. But in eukaryotes, the enhancers and silencers are much more complex. So we don't want to just say it's only binding to the promoter. We can say it's a binding to any regulatory region that's important for transcription. So what is a regulatory region? It's kind of like a promoter, it's just somewhere maybe close to the gene, maybe not, but it's what's participating in this DNA protein interaction. And you can have transcription factors that increase transcription. Or inhibit transcription. This is really important when we start thinking about the genetics behind gene regulation. We can discover mutants where a gene is never expressed. That probably means that there's a transcription factor upstream at the protein. So the broadest term to control rate, how do you control rate? You can speed something up, you can slow something down. And you do it through these DNA protein interactions. So this slide is another figure from your book, just to get at you on this. So we have that initiation of the sigma factor helping bind to the promoter. And then the RNA polymerase just keeps going from this 5' to 3' direction. And what we said is you do not need an RNA primer. So you do not need primase. So primase was a really important player in DNA replication. You don't need that for RNA elongation. RNA polymerase is already working with RNA. You don't need primase. So again, there's not much information on the slide because it's the same process we've already talked about, building in that 5' to 3' direction. This slide, just again to kind of hopefully hammer home the fact that the directionality depends on the orientation of the DNA. Right? The RNA is going to be synthesized anti-parallel. So this is the exact same thing we saw last time. Certain genes are being transcribed using the bottom side of this template, and others are being used at the top of this template. The template is what is going to act as a complementary strand for the RNA. We have to use this term a lot, but what's the non-template? What's another name for the non-template strand? Your book kind of uses these words interchangeably. I use them interchangeably. It's really to draw emphasis. If we're thinking about transcription, I want to think about the template strand, but the reason it's the template strand is because there's another complementary strand. Coding strand, right? So that's where we're heading. We're heading towards translation. This mRNA is going to provide the triplet nucleotide code that's going to translate into amino acid sequence. Any questions on that? So this is pretty quick, I know, because you don't have to memorize these details. What you need to do is be able to identify strands and their polarity, think about what's being transcribed, and then think about how that's going to be turned into a protein. The genetics come at, well, what happens if we change this sequence? Is the protein altered? Is transcription altered? So you don't have to memorize the mechanism, but it might help you give you context for when you start adding genetics kind of on top of it. Okay, so here's another example that's really E. coli specific for termination. And the reason I included it up here is because it shows kind of, again, why protein DNA interactions are so important in mediating all of these processes. So this is a very specific type of termination called row dependent. So this is not universal, but it kind of shows you a couple things. So the first part is showing you that there's a DNA sequence that's specific. So this is called the RUT site. So that's the row, that's for row utilization. So on the right-hand side, it's that little yellow thing. It's part of the DNA, but it also gets transcribed as part of the RNA. So it's a recognition site. It's a specific sequence of DNA that a protein is going to come and interact with. So then the polymerase is the big brown thing, right, that we've seen kind of going through all of these slides. The little purple thing is the row protein. So I'm not going to try to, so row is like this, right? That's the Greek letter row. So it really acts as it's almost like a helicase. So it's keeping the DNA-RNA hybrid from interacting, right? This RNA, single-stranded, has an affinity to try to interact with other aspects or other parts of the single-stranded DNA. So in its normal configuration, this row protein is trying to keep them apart. Right, it's trying to keep apart the DNA and RNA hybrid. And it acts at that as that function until it gets to the recognition site, or that RUT site. So it eventually catches up to the polymerase. So that's what's shown down in this third right here. Catches up to that polymerase, right? There's also in the termination site, there's something that makes that stem loop structure. So that stem loop structure physically allows the row protein to be close to the polymerase, and then they disassociate. So do you have to remember row independent termination? No. It's just an example of how you need certain protein DNA interactions for initiation, elongation, and then here we'll see exchange. Any questions on that? So the sequence is in the DNA, it has to be transcribed, it will not make it into the protein. Where the protein is going to end is going to be dictated by another sequence that we haven't thought of yet, a stop codon. But RNA, if you remember, we said has a primary and a secondary structure. Because it's single-stranded, it still has all of those bases that are looking to make base pairs. So here it's making some of the secondary that allows physical interaction between Rho and polymerase. That's kind of all that. So that was all prokaryotic specific. A lot of that carries over to eukaryotes, but there's some really important differences. So what are some of the differences that might affect transcription in eukaryotes? So prokaryotes usually are a single cell. So what are all the things about eukaryotes that are going to affect how we think about transcription? Any thoughts?
The size of it?
Yeah, I think so. They're larger cells and more complex cells. What's a consequence of having these larger cells? You have a nucleus. Prokaryotes don't really have a nuclear membrane, right, that separates from the cytoplasm. That's going to be really important for how we treat RNA before it gets translated. Larger cells, what else? Do they usually occur singularly or in multicellular things? And if you're multicellular, usually you have cells that have specific function, right? You have kind of division of labor. So this is the really important part. Multicellular adds the requirement that a gene is transcribed in the correct type of cell. And at the right time. This can be either development or in response to environmental stimulus. Right? A prokaryotic cell, it responds to its environment irrespective of the cells around it, not always true. But, right, your liver has to do something different than your kidneys and different than your brain. The genome is identical. The DNA in all of these cells is identical. What's different are the expression of transcription factors, which are going to then feed into the expression of specific genes. So this is where we're going for eukaryotic transcription. Right? In some scenario, whether it's development, a certain tissue, response to an environmental stimulus, gene A is being transcribed and translated, gene B is off. We can have scenarios where the opposite is true. We can think about examples where this is really important, right? So in a muscle cell, you don't want to express insulin or hemoglobin. And why didn't you want to, right? You have limited resources, it could cause disease or it could cause malformations. So want to is a weird way to say it, but basically the muscle cell would not function properly if it was expressing the gene, the insulin gene or hemoglobin. Pancreas, you want one but not the other, right? Beta cells of pancreas, you want insulin on, not hemoglobin. And red blood cells, immature red blood cells, right? Mature red blood cells do not have nucleus, they're anucleated. Immature red cells, you want hemoglobin. That can only be accomplished through transcription factors. The DNA is identical for all of these cells. What's different is the expression of transcription factors. So what did we say transcription factors were interacting with regulatory regions, right? So there's two components of a regulatory region. There's the core promoter. Most genes in your genome, if they were kind of lacking a promoter, would have very low levels of expression of promoter is necessary to control regulation and actually increase transcription. And then there's enhancers. So these are the types of regulatory elements. So enhance is like increasing, we talked about that earlier. That's stimulating transcription. And silencers. So as their name implies, they're inhibiting transcription. This is already information we wrote previously. I just am now giving them names, specific names. Because in bacteria, we don't really think of transcription regulatory regions the same way we do in eukaryotic genes. So in eukaryotic genes, we have the specific names. So eukaryotic genes are really complex. So we're not going to go into a lot of the details, but I want you to think about this. It's not just a single regulatory region. It's not a single DNA protein interaction. It's this really complex protein interaction. And here the regulatory regions can be really far from the transcription start site. So in bacteria, we'll see everything's kind of clustered for efficiency. They have really small genomes. Eukaryotes are not the same. So you don't have to remember tau boxes or anything like that, but just look at the bottom. Look at all the proteins that are necessary and the contortion of this DNA molecule to activate transcription. So the key thing here is that regulatory sequences can be really far away from each other. I know my handwriting is always messy, but I feel like as the semester's gone on, it's gotten worse and worse and worse. So there's one last aspect of eukaryotic regulation that I want to talk about, and it really deals with what happens when you have two homologous chromosomes. So bacteria, right, have a single chromosome. Usually they have plasmid. Eukaryotes, you have those two homologous chromosomes, and this is going to be really important when we do genetics. So it's this idea of the nomenclature doesn't make a ton of sense to me, like why it's named this way, but it's what we use, so we're going to stick with it. But it's kind of cis versus trans regulation. So what does that mean? How would we use this information to say something about the regulation? So cis elements, or we call them cis-acting elements, they usually affect transcription of genes that are on the same chromosome. So bacteria, we'll see they're really close. They're usually part of the promoter sequence or the operon. But in eukaryotes, it's not the same, right? In eukaryotes, we have this regulatory promoter that's really far away from the transcription start site that interacts with the bending action. But that would be a cis-acting factor. It's only going to affect the genes on the same chromosome, even though it's not physically close. So by contrast then, the trans-acting factor can affect anything kind of in the cytoplasm that it code occurs with. So I guess I'll say any below there. So what does that mean? It means it could be really far away from the gene of interest. It could be on a separate different chromosome, but it's mostly going to be a protein, right? So not all of them, but these are often going to be proteins. So in that example here, that mediator and all of the TFI proteins, they're going to be trans-acting factors. They were not transcribed and translated as part of this chromosome. They were translated and transcribed somewhere else. They exist kind of in the nucleus of the cytoplasm and come to this gene location. So that's going to be really important. If you have a mutation in a trans-acting factor, your phenotype is very different than if you have a mutation in a cis-acting factor. Any questions on that? I know it's kind of just definitional at this point. We won't see these in terms of genetics until after the exam, but it's really important to see the build on it. Any questions? Okay. Let me look at topic. So there's a question, what assignments will we have next week? That's a good question, I'll get to that at the end. So this is where we were supposed to finish last week, and so all this new stuff is kind of new material with your new reading. So hopefully you already saw system transaction factors for your reading last week, all this new stuff you're reading up yesterday. So RNA processing. So in eukaryotes, we can't just make RNA and have translation act immediately. That's what happens in prokaryotes. In RNA, in eukaryotes, we need this RNA processing. How many people discover RNA processing? It's really kind of interesting. For a long time, you might have been taught to say, well, the central dogma is kind of like one DNA, one gene, one protein relationship. People started understanding that wasn't true, and that a single DNA strand, a single gene could result in many proteins called isoforms. So people were doing these experiments kind of in the 60s and 70s, not very long after the structural nature of DNA was actually published, but they were seeing interesting patterns. So they found some really cool viral systems where they could, viruses usually integrate the DNA into a genome, and then they transcribe it. So they were taking RNA extracted from different parts of cells, and then making hybridizations with the DNA. And what they were seeing were these loops. There were parts of RNA that weren't, and vice versa, there were parts of the RNA DNA hybrid that weren't having a direct match. That meant there was extra material. And what they were seeing is that the RNA kind of outside of the cytoplasm was different from the RNA in the nucleus. So that suggested that RNA processing was occurring. So finding that phenomena is what we call splicing. That led to a Nobel Prize in 1993, but not for this woman. Right? She's the one who did all the experiments. Of course, she didn't get the Nobel Prize. Right? Two white guys got the Nobel Prize. So if you want to read more about the story and kind of the women behind the discovery of splicing, there's this link. It's really, really interesting. She went on to have a really great career, but not a Nobel Prize. So why do we care about splicing? We care about splicing because genes often have really complex structures. In bacteria, that's not the case, but in eukaryotes, it is the case. So there's many things. We're going to deal with things called untranslated regions. We have exons. We have introns. We need a start site. We need a stop codon. There's a lot of structure in genes, right? And these introns actually give us the ability to make alternative products called isoforms. So these are all terms that you should be familiar with, or at least in practice, as you start talking about them and being able to identify. So another specific aspect is that, this mRNA has to make it from RNA and mRNA, and it's passing through that nuclear residue. So a lot of the rest of, not exons, introns, those are kind of properties of the RNA itself, but things that we're going to add on top of the RNA or to facilitate that passage through the nuclear membrane to where the ribosome is. So this really challenges kind of what people for a long time dealt with, the linear model of DNA. So the DNA is going to be transcribed directly into an RNA molecule, it's going to be directly translated. Instead now we have what we call this collinear model, or sorry, non-collinear model. So it's not that a single DNA leads to a single RNA, it's just so this isn't the best diagram for this, but this just diagrams kind of the experiments that were done in the first kind of introductory slides. So here are some of the regions that we start seeing. You don't have to think about the Shine-Dalgarno sequence. When we have this RNA that's going to have now untranslated regions, start and stop codon. So we have our first clicker question. I think I wanted to just ask first, what does untranslated mean? If you just hear that, what's the first thing kind of it invokes? kind of what we've been talking about, right? That not all the RNA molecule makes it to the protein. So what's defining this region? This is your clicker question. So we keep seeing this basic model, right? Last time we saw it was coding region and regulatory region. Now we keep adding to it. We keep adding a little bit of complexity because that's necessary to think about the realistic aspects of transcription. So if the question's not clear in the clicker, what does untranslated mean? It means it's in the RNA, but it doesn't make it to the amino acid. And then the clicker question should be, well, what's defining that? So your options are the fact that it's not transcribed, so that's, think about what that means. Start codon, D and C are all of the above. So currently the answers are all over the place. So does someone want to defend one of their answers? Yeah.
I think it's all three because it's before the start codon, and then after the stop codon, and then that's.
Yeah, so I think that's some good logic. So I agree with the first two things that you said. That's kind of what's being shown in this figure. The protein coding region is being defined by those two codons, which we haven't defined yet, a start codon and a stop codon. So yes, totally agree with that. I don't know if I agree with the first part. So A is they are not transcribed. Does someone want to comment on that, share any ideas about that? Yeah, like you're raising your hand, not up high enough yet.
It has to be transcribed into mRNA.
Yeah, so that was kind of a tricky part of that question. So I think your intuition's good, but it's what's the definition of transcription? So transcription is from DNA to RNA. So if this is the RNA, they're in the RNA molecule, they're just not getting to the next step of translation. So I think, I like how you thought that through, but that is kind of a definitional thing that we have. So transcription is DNA to RNA. This is in the RNA, it's just not making it to that next step of the amino acid. That's translated. Okay, so the best answer here would be D. So it's really defined by the start and stop codon. So we want to maintain kind of, we have to maintain the protein coding region. Sometimes we want to maintain the untranslated regions because they have regulatory sequences, but we still need to make mature mRNA. So that's the end goal. We're still kind of thinking about how to process this into something that will be translated. So these are all the modifications we're going to see, and we'll kind of go through them a little bit. So the first, this is a different figure from a different book that I like a little bit better. Because it shows the coding and non-coding strand, and then the template strand has that exon introns kind of labeled, and then you can follow some of the color. So we're going to make a pre-mRNA that's going to have everything. It still has introns, it still has untranslated regions. We need to add a cap to the 5', so that's that lining. You'll see that. It's a very specific lining we added. Then we splice, and then we add a tail. That's not actually the order that it occurs in, but I think they wanted to show that you could explicitly see splicing. So splicing is the removal of introns. I think I've said splicing several times, but not really formally defined. So this is our first modification, this 5' cap. So this is really regulating nuclear export. So it's preventing degradation as well from exonucleosomes, right? It's identifying this mRNA as something that hasn't been translated yet. That's the key thing. There's a lot of RNA that's in a cell. There might be viral RNA, bacterial RNA, and a eukaryotic cell. There might be RNA that's already been translated and needs to get degraded. A lot of regulation of RNA happens to do that. So there's proteins waiting to attack RNA. The cell needs to tell those proteins to stay away, and one way to do it is through this type. So this is what it looks like kind of chemically. You have this modified guanine here, right? So this is our sugar. that's our base. So it's called a 7-methylguanosine. So we were dealing with prime carbons within the sugar, within the base, they're non-prime carbons. So if you find carbon 7, it now has a methyl group. Those aspects are important, but I do have a clicker question related to this that I think is important. You've been seeing kind of nucleotides joined in a very specific way. So what's different about this bond specifically? So what do you notice looking at this bond? Someone answered, they're looking at the phosphoruses. That's a good one place to look at. What about the phosphoruses? Or the phosphates? It does have phosphates. All nucleotides have phosphates. All nucleotides are making phosphodiester bonds. So there's something else that's happening here. Someone put it's attached to 2 carbons. I think that's a good way to start thinking about that. So how do we normally connect nucleotides in a DNA or RNA molecule? What's our mantra? In normal nucleotides, we connect 5' to 3'. So this would be the 5' here connected to the 3', right? 5' to 3'. But we're building in a very specific direction. The difference in this bond is it's a 5', 5'. So here's a 5' carbon, here's a 5' carbon. It's a fundamentally different way of connecting nucleotides. That acts as a very important signal. You don't see this in all the other kind of nucleotide bonds that we're dealing with. This functions as a signal along with that methyl group, right? So that's what I was getting at. So if you're, I think someone said it's attached to carbons. What about those carbons? They're both 5' carbons. So will you have to memorize this? No, but you should be able to think about identifying carbons, right? With the nucleotides. That's the fundamental principle. It's important for everything else we've been doing. Okay. So we have a 5' cap, we're going to add a poly-A tail. So what does poly-A mean? It just means a string of adenines, so a long string of adenines. So these can vary in length. It really is dependent on the gene, and there's some signals in there. You know, it can be as many as 250. And so that's what's shown kind of at the bottom. It's just a really long string of A's. There's a sequence within a gene, so the consensus sequence that tells a protein kind of that there's going to be a poly-A added later. And that's added kind of similar to the way we've always done it, 5 to 3'. So that consensus sequence is not found in all genes, but if you delete that from a gene, you don't get a poly-A tail. So it's kind of something that we know is really important for this poly-A. But why it's not essential in all genes is still an open question for research. So what is this doing again? It's really helping degradation. So the first one was important for nuclear export and degradation. This one's mostly just for degradation. So the only thing we haven't done yet is splicing. So often I'm asked this question, if introns are going to be spliced out, why do they exist? Does anyone have any ideas on why introns exist?
Regulation.
Any other ideas? Why is a really hard question. So the question I would like to actually think about is, well, what is the consequence? So why questions can be hard, but consequences we can think about, right? So I I agree, there's a consequence. We can regulate genes specifically with introns. Introns often carry regulatory reasons. That adds another level of regulation. Any other, what might be another consequence? We talked about a couple of them. We talked about isoforms. So those are different proteins from the same gene. I'll just say from the same DNA sequence. Yep, so that is how you get isoforms, yep. We'll see a couple examples of that. One other thing. You can get new genes. And can be acted on by natural selection. So introns don't exist to make evolution happen, to make regulation happen. It's a consequence. So if we have alternative splicing, it usually is producing certain isoforms. Sometimes things can happen, mutations can happen. We bring together exons that were never together before, that's called exon shuffling, and we get new genes. A lot of them will go away because there is no benefit of having them, but sometimes they will have a fitness advantage in evolution to have them. So those are the main consequences of having them. Genes are really variable in the number of introns, the size of introns. What's kind of the optimal size, I think, is really an interesting question. I don't know if there will ever be an answer. So these are just two examples of two different genes. So you have kind of this albumin gene that has a lot of these really small introns, or excuse me, a couple small and a couple really large introns. So here the colors I think are reversed, the light green is the intron. And then you kind of have that cytochrome gene that looks pretty regular, right? All the introns are the same size. What is the different properties of these genes that might dictate the size of these introns? But either way, often the point of this slide is that the coding region, the dark green, is often interrupted by many. How does a protein know where to splice? Again, it's all about the DNA protein interaction. So this is in the RNA sequence. So this is a consensus sequence that's in the RNA molecule now. It has to be in the protein sequence previously. So you don't have to remember the sequence, but there are consensus sequences that make very specific structures. So when an intron needs to be spliced out, it's a physical removal of that RNA sequence. So how do you remove things? You kind of cut them out. So this is what this diagram in your book is showing. And it's really, there's a lot going on. I'm not going to talk about all of it, but basically at the consensus sequence of the 5' a break is made. A physical kind of separation of the RNA molecule that allows kind of that lariat. So this is what they're referring to as the lariat. Like a lariat, right, is a rope that has a loop at the end. There's a physical interaction now within this molecule. That's what's so interesting about RNA molecules. They have secondary strength. They can kind of bend and twist in lots of different ways. And then it's removed. And then something brings the two exons together. So this is trying to show you the lariat, if you saw this figure in the book. And I think what's really important to remember is this is all happening within the splices zone. So it's a combination of proteins, right? It's not a free-floating DNA molecule. It's happening within there. So when they show kind of that exon existing in space, like by number 4, that's still being held in place by a protein. So if you saw this in the book and we're thinking, well, how is this occurring in cellular space? It's not. It's occurring within this context of this place. So this is your book's figure of all the different steps. So we have our DNA, we have transcription, we have all the regions that we need, the introns, the translated regions. The 5' cap is added here, right, number 2. Polyadenylation, and then splicing through those introns. Not all genes require a spliceosome. Some of them kind of can auto-catalyze introns. intron removal as DNA or protein independent, which is really interesting. So that's our end of splicing. So you don't need to memorize the details of splicing, you just need to think about the consequences. And think about how you would identify transcripts that might come from different proteins that might come from different transcripts. So don't memorize consensus sequences, just think about the processes that were involved, why they're important, so for nuclear export, for avoid degradation, and then splicing can result in different proteins from a single DNA. So what kind of RNA is this? So now we're going to just show different examples of how really important RNAs are being processed. So these are RNAs we're going to see next lecture in translation. So what type of RNA is this? So this is trying to help foreshadow. We're still going to go through RNA processing in different contexts, but also foreshadow what's happening in translation. So I'll leave this open. This is a tRNA. T stands for transfer. It's going to interact with the mRNA within the ribosome to transfer amino acids to the growing polypeptide chain. So there's a lot of things we've already seen in RNA. It has a secondary structure. It has this really typical clover-leaf secondary structure. What helps with the secondary structure? Right? Base pairing within these loops. They make stem and loops. There's a lot of other weird stuff that they have. They have this anticodon, which isn't weird, but it's important. We'll speak next time. But they also have these modified bases. There's like this symbol psi. It's an echo, it's a nucleotide, which is so this is just to emphasize that we think about the secondary structure a lot, which is the far right, but it actually has a 3D structure. And then it has the amino acid attachment point outside of that 3D structure. So we're going to really care mostly about the anticodon. We're not going to care about the other aspects of the secondary structure, but this is not how this molecule is transcribed. This is what happens as a process of RNA processes. So for the two people who care about chemistry, these are what these other bases look like. I don't think I kept the top half question. What is ribose was the question. Where is it attached? We're not going to do the top half, but think about that. This is what this process looks like. So they're calling it here a precursor to tRNA. So that would be the RNA that was transcribed initially, and then you get to the mature tRNA on the end. So what are we seeing? We're seeing at the light gray, we're seeing those untranslated regions. We're seeing a big intron down here, right? So there are consensus sequences within even tRNAs that need to get spliced out. So it doesn't look exactly the same that we saw kind of from precursor to mRNA, but there are some similarities. Processing has to occur to shorten the molecule, to bring that anticodon into that loop that's on the bottom, and that's done through splicing. Any questions on that? So I think we're going to fix the end early, which is good. You can ask me whatever, any questions. Hopefully this will catch up so we can just do translation now. So this is the other main RNA we're going to see on Thursday, this ribosomal RNA. So the R stands for ribosomal. They also have secondary structure. But the ribosome is a big complex of lots of RNA and protein. So that's where you can kind of start seeing in bees, like this really big three-dimensional space. It has a lot of different active catalytic sites, and some of the catalytic sites are due to RNA. So that's another aspect of RNA we didn't really talk about, but I think is just really interesting. We often think of something catalyzing as a protein, right? Enzymes catalyze reactions. RNA can actually catalyze chemical reactions. RNA, our RNA, ribosomal RNA, there's hundreds to thousands of copies in your genome. And you have to make ribosomes all the time. So this table from your book is just trying to show the total number of RNA molecules and proteins that are necessary to form these different subunits. What I want you to take home from this is that how are you going to transcribe, how are you going to regulate the transcription of all these rRNAs? You need to feed them into making ribosomes. You need a lot of them. What are some solutions to do that? Any ideas, thoughts? How are we going to make a ton of rRNA whenever we need it? Yeah. We need promoters. They can be really strong. What's another way we can increase transcription? So RNA, so I like that idea, thank you for sharing that. This has to happen in bacteria too. Bacteria, so the bacteria have promoters, but oftentimes they don't have, like in eukaryotes, we'd say strong would be like an enhancer. Bacteria don't have enhancers, but I like that idea. What's another idea? If you flip to the slide ahead, and I'll give you a hint, or maybe not. What's happening in this? It's not labeled, but can you think about what's happening? Can you figure out what's going to happen here? Maybe in top hat chat someone said something. Nope. So this is an array. What that means is tandem duplication. I'm going to switch back to the other slide real quick. You can increase copy number. So when we did kind of chromosomal mutations earlier, a couple weeks ago, we said that lots of duplications and deletions can have consequences on human health because you're taking a really important biochemical pathway and disrupting it through disruptions in dosage. RNA are not like that. You need as many as you can get all the time. So you can actually just increase copy numbers. So that's what's shown here. So in the black are the structural part of the gene, and then the green is kind of like a spacer, it's a sequence in between. And then in B, it shows you the codons are kind of the raised up part, and the introns are the lower part. So we've talked about how most of the time, genes are different orientation, and there's really strong areas that limit transcription. That's not the case with these rDNA arrays. One polymerase can just chug along and make tons and tons of RNA, and then another one can start immediately after it. So by having this increased copy number, you can transcribe a ton of rRNA rather than one. So that was the point of this. We've seen tandem duplications before. We talked about them often being deleterious. Here they're essential for rRNA. So these RNA also have to be processed. So this is what processing looks like in RNA. And it's a little bit different for different subunits. So this is a figure from your book. So it has kind of prokaryotic and eukaryotic versions. Most of the time in prokaryotes, we don't talk about splicing because they don't have introns. So the way that they're generally dealing with this processing is relying on those methyl groups. So are eukaryotic genes. They are still splicing things out, but it's really interesting how rDNA are universal. Everything has a ribosome. How you process something that's so universal, you actually kind of move away, take a step back from those spliceosomes and rely on DNA modifications through these methyls. So again, you don't have to memorize this process, but just think about all the different molecules that have to be processed to get us to translation. We have to process our primary transcript through 5' cap, polyadenylation, splicing. We have to process a tRNA and a ribosomal RNA. Okay, so that was my last slide. I'm going to answer whatever questions you have. There was also a question in, Top Hat about what assignments will we have next week. So I'm gonna try to